The Cell Cycle, Mitosis vs. Meiosis, by Carrie Donahue and Kate Warren. Mitosis and meiosis are essential processes that help our body both produce and replenish our cells. In this presentation, the differences and importance of mitosis and meiosis will be discussed. Mitosis occurs when a diploid cell with two sets of chromosomes divides in half to create two daughter cells that are genetically identical. Before mitosis actually occurs, however, an essential part of the cell cycle called interphase must occur. Interphase is by far the longest phase of the cell cycle and consists of three different phases, G1, S, and G2. During G1, the cell grows and prepares for its chromosomes to duplicate, which they do in S phase. During G2, the cell grows even more as it prepares for cell division. Specifically at this point, the cell has two copies of centrosomes, a nuclear envelope, and nuclei still intact. Now the cell is ready to divide as it enters into mitosis, or the division phase of the cell cycle. The first phase in this cycle is prophase. In prophase, each replicated chromosome contains two sister chromatids. Chromatin fibers within each chromosome condense, the nucleoli disappear, the mitotic spindle begins to form from microtubules and proteins, and the centrosomes attached to the mitotic spindle begin to move apart. In the next phase, called prometaphase, the nuclear envelope starts to decompose, the chromosomes continue to condense, and each sister chromatid contains a kinetochore, or proteins associated with specific sections of DNA. In this phase, some of the microtubules from the mitotic spindle attach to kinetochores of sister chromatids, forming kinetochore microtubules. In metaphase, which is the longest stage of mitosis, the centromeres of the replicated chromosomes, or area where two sister chromatids attach, line up along an imaginary plane called the metaphase plate. Each replicated centrosome is now at the opposite pole of the cell, and the kinetochores of each sister chromatid are attached to the microtubules from each pole. During anaphase, the shortest phase of the cell cycle, cohesion proteins that connected each sister chromatid to one another are cleaved, and they detach. Each sister chromatid now moves towards an opposite pole of the cell as their kinetochore microtubules shorten. In telophase, two different nuclei form within the cell, each with its own chromosomes. The nuclear envelope and nucleoli begin to form again, and the chromosomes decondense. During cytokinesis, the cytoplasm divides, forming two distinct cells, though each is genetically identical to the original cell. In animal cells, this is called cleavage furrow. In plant cells, however, a cell wall forms between the two cells.
Though meiosis is similar to mitosis in many ways, the two are also very different. Meiosis, unlike mitosis, produces four genetically different haploid sex cells. Though the meiosis cycle includes interphase, there are other significant differences between the two processes that should be noted. During interphase of meiosis, a homologous pair of chromosomes in a diploid parent cell replicate, forming a homologous pair of replicated chromosomes, or a diploid cell. During meiosis, there are two different cell divisions, meiosis 1 and meiosis 2. Each cycle consists of prophase 1 and 2, metaphase 1 and 2, anaphase 1 and 2, and so on. During prophase 1, chromosomes start to condense, as in mitosis, and the nuclear envelope begins to fragment. One important difference between meiosis and mitosis, however, is that during prophase 1, crossing over occurs. This is when two non-sister chromatids held in synapsis exchange segments of DNA. Now, each homologous pair has several chiasmata, or points where crossing over has occurred. The spindle fibers begin to spread apart, and the homologous pairs move towards the metaphase plate. During metaphase 1, the pairs of homologous chromosomes line up along the metaphase plate, and the chromatids of each homolog attach to kinetochore microtubules. During anaphase 1, proteins that held together the homologous chromosomes are cleaved, and each homolog, attached to the spindle apparatus, moves towards an opposite pole of the cell. At the start of telophase 1, each pole of the cell has a haploid set of replicated chromosomes with sister chromatids containing DNA from other sister chromatids. After cytokinesis occurs, two haploid daughter cells are formed. Now, another part of the meiosis cell cycle occurs, or meiosis 2. No replication occurs between meiosis 1 and 2, and meiosis 2 is extremely similar to regular mitosis. In prophase 2, a spindle apparatus begins to form within each of the two cells, and chromosomes move towards the metaphase plate once again. In metaphase 2, the genetically different replicated chromosomes line up along the metaphase plate, and kinetochores of sister chromatids attach to the spindle. During anaphase 2, the sister chromatids split and move towards opposite poles of the cell. In telophase 2 and cytokinesis, each cell already formed in meiosis 1 splits into two more cells, making a total of 4 haploid daughter cells. Each of these cells is genetically different from the parent cell. Though these two cycles are very similar, prominent differences exist between them. The biggest one is what these cycles actually accomplish. For instance, mitosis makes all types of cells in our body except for sex cells, while meiosis yields all of the sex cells, or the eggs and the sperm. Even though they are different, both mitosis and meiosis are essential processes that make up all of the cells in our body. Without these cycles, cells and reproduction would not occur, and life as we know it would not exist. By further studying these processes, and especially genetic inheritance in cells produced by meiosis, we can learn even more information about how our body works.